Welcome back to BioMindscape. Today we are going to know about the biomolecules. So what is a carbohydrate? They are the one of the most abundant class of biomolecule in nature, and they are found widely distributed in all life form. Chemically, they are aldehyde and ketone derivative of a polyhydric alcohol. Major role of carbohydrate in living organism is to function as a primary source of energy. These molecules they serve as an energy store metabolic intermediate and one of the major component of bacteria and plant cell wall, and also these are the part of the DNA and RNA which you will study in the next chapter. So the cell wall of the bacterial plant they are made up of polymer of carbohydrate. They also act as an informational material and they link to the surface of protein and lipid to act as a cell-cell interaction, and in the interaction between the cell with other element in the cellular environment where they play a role. The classification of carbohydrate is that they are found in various forms ranging from a simple sugar to a complex polymer, and they form of one unit according to they have been classified. They are commonly classified into three categories, namely monosaccharide, oligosaccharide and polysaccharide. So what is a monosaccharide? They are simple sugar which cannot be further hydrolyzed into simple form. These monosaccharides contain free aldehyde, that is CHO, ketone, that is C double bond O group, which with two or more hydroxyl, that is OH group, with a general formula of CN in bracket H2O bracket close N. So this is the formula of the general formula for which compound? Monosaccharide. So based on the number of carbon atom and functional group, monosaccharide are classified as triose, tetrose, pentose and hexose. These are the classes of monosaccharide based on the number of carbon atom, and the class of monosaccharide based on the functional group they are like aldose, glyceraldehyde, erythrose, ribose and glucose, whereas for ketose it is dihydroxytone, erythrose, then ribulose and fructose. What is an oligosaccharide? Oligosaccharides are carbohydrate which have 2 to 10 unit of monosaccharide. They are joined together by glycosidic bond. Some commonly occurring oligosaccharides are maltose, lactose, sucrose, etc., which are disaccharide, means they are having two or more saccharide, two monosaccharides. Then polygosaccharides are polymers of more than 10 monosaccharides unit. They are joined together by glycosidic linkages, and they are classified in a number of ways depending upon the type of the repeated monosaccharide unit, like homo and heteropolysaccharide, in the degree of branching, and in this type of glycosidic linkages between the monomeric units. Example of some common polysaccharides are starch, glycogen, then cellulose and chitin. Carbohydrate can be conjugated to protein and lipid to form the glycoconjugate, and these are the three type of glycoconjugate: the glycoprotein, then proteoglycan and glycolipid. If the protein component is predominant in association with the carbohydrate and protein, it is called glycoprotein. So if it is associated containing major amount of carbohydrate, then the protein, then it is known as the proteoglycan, and when the carbohydrate conjugate with the lipid, it is known as glycolipid. So there are various structure and property of carbohydrate. For monosaccharide, the structure of some common monosaccharide are given in the figure. Monosaccharides such as the glucose exist both as straight chain structure and cyclic structure, whereas the cyclic structure are the result of hemiacetal formation by the intermolecular reaction between the carbonyl group and the hydroxyl group. All the monosaccharide except the dihydroxy acetone contain one or more asymmetrical the chiral carbon atom bounded by the four different group, thus are optically active isomer, that is enantiomer. A molecule with n chiral center has two second stereoisomer, thus the glyceraldehyde with one chiral center has 2 is equal to 2, that is glucose with 4 chiral center, that is 2 raised to 4 is equal to 16 stereoisomer. So the orientation of the OH group that is most distant from the carbonyl hydroxyl group is on the right side of the carbon atom, that bearing it the sugar D isomer, and when it is on the left, the sugar L isomer is there. So most of the sugar present in the biological system are D sugar. Isomeric formation of monosaccharide, that different only in their configuration. There is only difference in their configuration, but the hemiacetal, it formed due to the reaction between the alcoholic and aldehyde group of monosaccharide, or hemiketal form due to the reaction between the alcohol and the ketone group of the monosaccharide. So the carbon atom, they are known as enantiomer. The carbonyl carbon atom is known as the anomeric carbon. So in the alpha amine anomer, the OH, that is hydroxyl group of the anomeric carbon, is on the opposite of the sugar ring from the CH2OH group at the chiral center, that designate the dextro and the levo configuration, that is C5 in the case of the glucose. That means here the 5 carbon atoms are present, and the other anomer is the beta anomer. So the interconversion of the alpha and beta anomer is aqueous solution. It is known as mutarotation. So in which only the one ring, that is formed briefly into the linear form, then close again to produce the beta anomer. So there are two cyclic form of glucose, alpha and beta 1. So isomer have different configuration of OH only at one carbon atom, and they are known as the epimers. The most important epimer of glucose are mannose epimer at 6,2 and galactose epimer is at 6,4 as shown in the figure. Then oligosaccharide, what are they? They consist of 2 or 10 monosaccharides joined by the glycosidic linkages. For example, disaccharide, the maltose contain 2 D-glucose, that is dextrose glucose residue, joined by the glycosidic linkages, which is a covalent bond, which is formed by the joining of the hydroxyl group of one of the monomer with the anomeric carbon of the other sugar unit. So lactose is made up of 10 galactose and dextroglucose residue. The disaccharide can be hydrolyzed to yield their constituent monosaccharide by boiling with the dilute acid. It forms the hydrolysis of the sucrose yield at a mixture of glucose and fructose. Other oligosaccharides are trisaccharide, for example raffinose, which is made up of glucose, galactose and fructose unit. Then tetra, penta and hexasaccharides are there. What is a polysaccharide? Depending on the functional role played by the homopolysaccharide, they are divided into storage polysaccharide and structural polysaccharide. The storage polysaccharide and serve as a storage for the monosaccharide that is used as a fuel. So starch is an example of storage polysaccharide in plant, and glucagon, it is a storage polysaccharide in animal. So the structural polysaccharide such as the cellulose and chitin, it serve as a structural element in plant cell wall and animal exoskeleton respectively. Then heteropolysaccharide, unlike the homopolysaccharide, it provides the extracellular support for the organism in the extracellular space of animal tissue, and these form a matrix that hold the individual cell together and provide a shape, support and protection to the cell and tissue. So the name and property of some homopolysaccharides are given here, like starch, glycogen, cellulose, chitin, inulin, then pectin, dextrin and xylene. Then the constituent monosaccharide alpha D glucose here, beta D glucose for cellulose. Then for chitin it is beta N acetyl D glucoxyamine. Then for inulin it is beta D fructose, whereas for pectin it is alpha D glucuronic acid. Then dextrin, it is alpha D glucose. For xylol it is beta D xylose. And the biological significance for it is given here. For starch it is storage of energy in plant. For glycogen it is storage of energy in bacteria and animal. And for cellulose it plays structural role in and provides rigidity and strength to the cell wall. It plays structural role and provide rigidity to exoskeleton and insects for the who gives, for example, chitin. Then inulin is storage of the energy in plants. Then pectin, it has a structural role that holds the cellulose fibrils together in plant cell wall. Dextrin, it plays structural role as an extracellular Adhesive in the bacteria and xylol, it has storage and supporting role in the plant. Example of some common polysaccharide are starch. Starch occurred in plants as a reserve carbohydrate in tuber, seeds, fruits and roots. So it is composed of two homopolysaccharide, amylose that is 15 to 20%, amylopectin that is 80 to 85%. Amylose is a linear polymer of alpha D glucose monomer and linked by the alpha 1 by 4 bonds. Amylopectin, on the other hand, it consists of a glucose unit that is linked by alpha 1,4 glycosidic linkages like amylose. However, unlike amylose, it has a highly branch. So, branch points occur every 20 to 30 glucose residue, and linkages at the branches point is alpha 1,4 glycosidic. So it is characterized by the blue color of the starch with the iodine is due to the amylose. So in contrast, the amylopectin, it gave only a dull reddish brown color with the iodine, and the enzyme present in saliva is salivary amylase, and the pancreas consists the pancreatic juice, pancreatic amylase. Hydrolysis of alpha 1,4 glycosidic linkages of starch therefore digested into the monomeric glucose residue. Glycogen, what is it? Extensively branched storage homopolysaccharide. It is found in animal, similar to the amylopectin, and it consists of a glucose unit joining together by alpha 1,4 glycosidic linkages and having an alpha 1,6 linkages at the branching point. The muscle cell, what does it contain? The glycogen at 1 to 2% of the dry weight, and liver cell, it contains 10% of the dry weight as a glycogen. What is the cellulose? It is the most abundant extracellular structure polysaccharide found in plants. So it is also the most of the abundant of all the biomolecule in the biosphere. It is a primary structure component of plant cell wall. Structurally, cellulose is a linear polymer which is up to 15,000 D-glucose unit linked by the beta 1,4 glycosidic bond. So unlike the starch, cellulose cannot be digested by human, as in human gut lack the beta 1,4 glycosidic bond. It hydrolyzes the enzyme known as the cellulase. So cattle are and termites, they can digest the cellulose as their gut harbors symbiotic microorganisms that secrete the cellulase, which hydrolyze and digest the cellulase. Cellulose, the animals can digest, but humans can't. Then chitin, what is the chitin? It is a linear polysaccharide of beta 1,4 linkages N-acetyl D-glucosamine residue. It is the main structural component of exoskeleton of invertebrate like crustacean, insect and spider, and the main component of cell wall of the most fungi is chitin. Chitin and cellulose have similar structure except of the hydroxyl group at the second carbon position of the cellulose. It is replaced by an amido group in the chitin, and extensive hydrogen bond of the N-acetyl side chain. It makes the chitin tough and insoluble polymer. Then there is a peptidoglycan. What is a peptidoglycan? It consists the rigid component of bacterial cell wall. It is a heteropolysaccharide of alternative beta 1,4 linkages and N-acetyl muric acid (NAM). N-acetyl muric acid, the N-acetyl D-glucosamine (NAG) residue. NAM and NAG residue, the linear polysaccharide chain, are cross-linked by a short peptide attached to the N-acetyl muric acid, and the cross-linkage by the peptide wield the polysaccharide chain into a strong sheet that develop the entire cell and prevent the osmotic rupture of the cell. Lysosome, which is an enzyme present in the human tears that kill the bacteria by the hydrolysis of the beta 1,4 glycosidic linkages. We know about what is an agar. It is a gelatinous heteropolysaccharide present in the cell wall of marine and red algae such as the Gelidium. Then you can name all the other organism or species of algae, red algae, that contain this agar. It is a mixture of sulfate heteropolysaccharide which is made up of dextrogalactose and levogalactose, derive ether link between the C3 and C6. Agarose is an agar compound with very few charge group, that is sulfate and pyruvates. It has a molecular weight that ranges up to 80,000 to 1,40,000. If agar and agarose are dissolved in hot water, they form a solution which upon cooling set and form a gel. So agarose gel are used as inert support for electrophoretic separation of nucleic acid. Agar is used for form of surface of the growth of the bacteria and plant tissue culture. Now, what is a fatty acid and a lipid? Lipid are a group of organic compounds that are found in living organism. They vary in the structure and function because they have a hydrophobic and non-polar natural nature. Lipid are soluble in organic solvent. Lipid are primarily made up of hydrocarbon chain that connect to the glycerol via ester linkages. So we broadly classify lipid into two categories, the simple and the compound lipid. Various types of lipid are included within the two major category of lipid. These include the fat, triglyceride, glycerol, wax, polylipid, phospholipid, then steroids, etc. So fatty acid they are obtained as a result of the hydrolysis of the fat. Naturally occurring fatty acid they are generally synthesized from the two carbon units and hence contain every number of the carbon atom. They synthesize from the two carbon unit. The fatty acid chain may be saturated having no double bond or unsaturated having only one or more than one double bonds. So the number of carbon atom in a fatty acid hydrocarbon chain start from the carboxylic carbon and ends at the methyl carbon in increasing order. For example, we can see the figure. Carboxylic carbon is designed as. So have you seen the figure? Carbonyl carbon, it is designed as 1 and methyl carbon in number 18. So fatty acid are denoted by total number of carbon followed by a colon and then total number of double bond with a delta having a superscript number which define the position of the double bond in parenthesis. For example, a fatty acid with 18 carbon and with 2 double bond between C9, C10, C12 and carbon 13, that is carbon 9, carbon 10, carbon 12, carbon 13 will donate as 18:2. That will be denoted as delta symbol 9, 12. So unsaturated fatty acid are of two types. Can you name them? Mono and polyunsaturated fatty acid. Mono means single, that means single unsaturated fatty acid, and poly means many. So many unsaturated fatty acid. Some of the example for monounsaturated fatty acid are oleic acid, whereas for polyunsaturated fatty acid it have linoleic acid, linolenic acid. 2 double bond for linoleic acid. Linoleic acid consists of 3 double bond and arachidonic acid contain 4 double bond. So we can see the various names, common name of saturated and unsaturated fatty acid. Lauric acid, myristic acid, palmitic acid, stearic acid, arachidonic acid. Number of carbon 12, 14, 16, 18 and 20 respectively. And unsaturated fatty acid, oleic acid, linoleic acid, arachidonic acid and palmitoleic acid. Here 16, 18, 18, 20. Palmitic 16 number of carbon, oleic acid 18, linoleic acid 18 and arachidonic acid 20. Let's classify lipid. It has simple and compound. What is a simple lipid? Simple lipid is a fatty acid ester which have various alcohol with no other group. So it have only alcoholic group, for example fat and wax. Triglycerol and waxes are example of simple lipid. Triglycerol, they are also known as the neutral fat. These are the compound, they are the ester of glycerol and fatty acid. So the triglyceride, they contain a small fatty acid in 3 ester position. They are called simple triglyceride. Triglyceride contain more than one type of fatty acid and its third position is known as the mixed triglyceride. So triacyl glycerol is essential for providing energy to the body and they are the vehicle of energy storage primary in the adipose tissue. Then what is a wax? Waxes is a form of result of esterification of fatty acid with the monohydric alcohol of high molecular weight. So they are found widely in various organisms, and wax serve as a protective coating on the surface of an animal and as well as the plants, and also reduce the water loss in tropical plants. Now what is a compound lipid? They are also a fatty acid ester with alcohols and contain an additional group. So fatty acid compound of this lipid the hydrophobic tail and the alcohol moiety along with the additional group, and it consists the hydrophilic head. Such a structure is known as amphiphytic molecule. Common example of compound lipid is phospholipid and sterol. Then phospholipid, they are amphiphytic molecule. They are made up of two hydrophobic fatty acid. For hydrophobic means water repelling fatty acid tails. You know about this, you have studied in the plasma membrane, the phospholipid, primary found in the cell membrane. So these compounds are composed of the fatty acid chain. They are attached to the glycerol backbone, glycerophospholipid or sphingosine based backbone. So a modified phosphate group, it occupies the 3 carbon of the glycerol in the glycerophospholipid, and the phospholipid, it is defined by the kind of modified attached to the phosphate group. So most common is choline, that is phosphatidyl choline, and serine, phosphatidyl serine, that is similar to the sphingolipid, where the head group contain only a hydrogen atom and it is known as the ceramide, and many a time the head group is sphingolipid can be phosphocholine yielding others phosphomyelin. So phospholipid, the fatty acid chain, it is attached to the sphingosine backbone by amide linkages instead of the ester linkages. So phospholipid do not spontaneously, it does not mix with the water. Instead they acquire a pear shaped structure known as the micelle. So what is a sterol? They are the distinct form of other lipid having a peculiar 4 fused ring structure. Likely other type of lipid discussed so far, steroid also have a hydrophobic nature, insoluble in water. In a cell, steroid act as a receptor ligand and help to control the metabolism. What is a cholesterol? Most common derive of steroid, synthesized primarily by the liver. Cholesterol is key precursor of all steroid hormone, including testosterone and estradiol. So beside cholesterol is also found in the plasma membrane of most eukaryote, and it provides the rigidity. Common example found in plant are phytosterol and stigmosterol, which regulate the membrane fluidity and permeability, and ergosterol is important precursor of vitamin D and it's typically found in fungi. Wishing you all a very happy new year and stay tuned to BioMindscape. Do like, share and subscribe.